Hello and welcome. Today you will learn about one of the most significant biological processes in life, which is cellular respiration. Cellular respiration is a process in which food energy is converted into chemical energy such as glucose into ATP. So without further ado, let's go and define our variables. So here are the beads that we will use for each element. The yellow will represent the sulfur, the black will represent the carbon, the white will represent the oxygen, 3 purple, 1 yellow will represent ATP, which in this case is energy for the cells, and the coenzyme A will be represented by red, yellow, and green. The FADH will be represented by 2 orange and 1 red. So in this case, this is supposed to be FADH2. And the NADH is represented by 2 light blue beads and 1 red bead. Here's the general structure of a mitochondrion. First of all, you'll see that it has 2 membranes, an outer membrane and an inner membrane. Between that is the intermembrane space. And you'll see that there's invaguations inside the inner membrane, and that's called the cristae. The fluid inside the inner membrane is called the matrix, and this is where the 70S ribosomes and the enzymes are kept here. A neat aspect of the mitochondrion is that it can self-replicate with its own mitochondrion DNA. Lastly, before we get into glycolysis, I want to do a quick overview of the process. When we ingest food, we have our glucose. This glucose is then converted into 2 pyruvates by glycolysis. The pyruvate is then sent down to the link reaction, in which it is converted into acetyl-CoA and moves down to the Krebs cycle. In the Krebs cycle, the acetyl-CoA is now reduced to FAD and NAD, which then can be transferred into the ETC, or the electron transport chain, to produce even more ATP. Although you do gain ATP from glycolysis and Krebs, majority of your own ATP is developed from the ETC. Hence, your net product is a total of 34 ATPs.
Glycolysis takes place in the cytoplasm of the cell. It begins with 1 glucose molecule, the formula being C6H12O6. Then it becomes glucose 6-phosphate through phosphorylation, which is the addition of a phosphate molecule to an organic molecule. This then becomes fructose 6-phosphate, and fructose 6-phosphate is then converted to fructose 1,6-biphosphate. 2 ATP are converted to 2 ADP. Then the fructose is converted to 2 triose phosphate molecules, and here NAD+ becomes NADH. Each triose molecule then becomes a glycerate 3-phosphate molecule, and each glycerate 3-phosphate molecule becomes a pyruvate molecule, and here ADP is converted to ATP.
In the link reaction, the pyruvate from the cytoplasm is converted into acetyl coenzyme A. This pyruvate right here just entered the mitochondrial matrix from the cytoplasm. Once in the matrix, the pyruvate is decarboxylated and oxidized to form an acetyl group. 2 high-energy electrons are then removed from the pyruvate. These react with the NAD+ to form and produce the reduced NAD. This is called the link reaction because it links glycolysis with the cycle of reactions that follow, and which produces the acetyl-CoA. In the Krebs cycle, the oxidation of acetyl groups is linked to the reduction of hydrogen carriers. Most of the energy released in the oxidations of the link reaction and the Krebs cycle is used to reduce hydrogen carriers such as NAD+ and FAD, and for every turn of the cycle, the production of reduced NAD occurs 3 times. The carboxylation occurs twice and the reduction of fat occurs once. In addition, one molecule of ATP is also generated in the process. Alright, so here we have the pyruvic acid or pyruvate from the glycolysis. This will then be converted into acetyl-CoA by the pyruvate dehydrogenase complex. Just a heads up, the dehydrogenase complex always forms NADs and FADs. The conversion to acetyl-CoA will form NADH in the process. Then acetyl-CoA will combine with oxaloacetate to form the citric acid, which is a 6-carbon. It will then lose a carbon dioxide but gain another NADH by converting it into isocitrate. This will be the same for ketoglutarate. However, once succinyl-CoA is converted into succinate, the conversion will produce an ATP. From succinate to fumarate, the conversion will then produce FADH2. The final NADH will be produced when malate is converted into oxaloacetate and the entire Krebs cycle once again starts over. Now let's quickly review its carbon process. From the 6-carbon citric acid, we see that it loses one of its carbon to carbon dioxide, and another one as a result to CO2. Then receive reduced hydrogen carriers in the process. At the end of the cycle, citrate is then reduced into the 4-carbon oxaloacetate.
The electron transport chain occurs in the inner membrane of the mitochondrion. First, the hydrogen carriers, which are NADH and FAD+, are oxidized and release high-energy protons and electrons. The electrons are then transferred to the electron transport carrier, which has several transmembrane carrier proteins as seen in the picture. When the electrons pass through the chain, they lose energy. This energy is then used by the chain to pump H+ ions from the matrix. The accumulation of H+ ions within the intermembrane space creates a proton motive force. Then, at the end, there is a lower hydrogen ion concentration in the matrix and a higher one in the intermembrane space. In chemiosmosis, the hydrogen ions move down the electrochemical gradient and diffuse back into the matrix due to the proton motive force. This is facilitated by the transmembrane enzyme ATP synthase. As the hydrogen ions move through ATP synthase, they trigger the molecular rotation of the enzyme, synthesizing ATP.
Thank you for watching and see you in the next video.